Hello, welcome to Mr. B's Biology. Today I'm going to be focusing on histology, particularly histology of blood. But first I wanted to talk about what it is we're going to be looking for. Formed elements. So formed elements are less than 0.5% of whole blood. If you're to take whole blood and break it up into its first two kind of major components, about 55% of that would be plasma and 45% of that would be formed elements. These formed elements are the cells, basically. So erythrocytes being the most common, roughly 99.9% of the formed elements you're going to be looking at are going to be erythrocytes or red blood cells. And then we have this whole list of leukocytes, white blood cells, eosinophils, neutrophils, basophils, lymphocytes, and monocytes, which actually become macrophages. And the last one here are thrombocytes, or platelets as they're also known as. Thrombocytes being really small cell fragments that were once part of a very large cell called a megakaryocyte. And that megakaryocyte basically fragments into these cell fragments, which are thrombocytes or platelets. I would love to do a whole thing about thrombocytes because despite the fact that they're cell fragments, they do some pretty remarkable things, particularly involved with hemostasis, but they also produce and release growth factors, platelet-derived growth factors, especially to help repair damaged blood vessels and even some of the surrounding tissue. Now, two terms I want to throw out here are granulocytes and agranulocytes. If you've already done blood, you're familiar with these terms. These are really important to help you distinguish what type of cell you're going to be looking at, particularly these different types of leukocytes. Granulocytes are eosinophils, neutrophils, and basophils. As you can see in my wonderful drawing here, they have these granules. They're peppered with these things inside their plasma membrane within the cytoplasm. And these things are secretory vesicles, basically, vesicles that are producing something. It could be a type of toxin. It could be something like lysosomes that contain digestive hydrolytic enzymes. It could be something like heparin or histamine that is going to be involved with an inflammatory response. They have lots of these vesicles that are used particularly for immunity, for immune defense. The agranulocytes will be our lymphocytes and our monocytes. They do have things in their cytoplasm. It's not that their cytoplasm is this void sea of nothing, but they don't have these thick vesicles like you see with the granulocytes. Okay? The other thing I want to point out here when we actually start to look at the slide images is that what you typically use when you do something like a blood smear is H&E stain. And you've probably seen this term, especially if you've already done a lab about this and you see on the side of that slide, it says something like blood smear, H&E stain, and maybe it has like a date when it was actually made. The H&E stands for hematoxylin and eosin. And hematoxylin and eosin is a very common stain that is used in a variety of processes. It's a very good general stain. If you want to get more specific, if you want to stain something like carbohydrates, you might use PAS, or if you want to stain really the DNA, you might use something like toluidine, some other type of staining process. But H&E does a pretty good general purpose of staining different types of cells within a tissue or even different tissue layers. Okay, so hematoxylin stains things that are acidic or basophilic. So particularly, it will stain the nucleus a dark purple or a dark blue color. Eosin, on the other hand, tends to stain things that are more positive or eosinophilic, and it will stain things more of a reddish or a pinkish, or in some cases, maybe an orange color. All right, so we have these kind of two contrasting colors. Hematoxylin staining basophilic substances, dark blue or purple, and eosin staining eosinophilic things, more of a pink, red, or even maybe an orange color. And this is why H&E is so good, particularly just in general staining processes of cells, because here you can differentiate the nucleus from the cytoplasm, or even in some cases, more specific cells like an erythrocyte that doesn't have a nucleus, so therefore it doesn't have any basophilic or hematoxylin stain components. It stains obviously red or pinkish. versus something like a basophil that has lots of these secretory vesicles that stain particularly dark with the hematoxylin. So with our formed elements, we can actually separate or differentiate based off of this very common, very basic H&E stain. So it is very good to kind of distinguish between these different cells. All right, just a quick overview. If you've done this already, great. This might be a nice little refresher, but erythrocytes, hopefully most of us are familiar with what erythrocytes do. Their primary purpose is obviously transporting gases, oxygen, carbon dioxide. Eosinophils, their primary purpose is involved with basically kind of an anti-parasitic effect in terms of immune defense, but they are also involved with inflammation. And in some cases, you might also hear it mentioned that you can also see these associated with certain types of cancer. All right. Eosinophilia is a term that is used to describe that you have higher numbers of eosinophils than you normally would. But obviously, context matters. Do you have eosinophilia because you have malaria, or do you have eosinophilia because you are suffering from chronic allergies or some chronic allergic reaction? So just throwing that term out there, you might have seen some of the drug advertisement commercials, or you might have heard this term. Neutrophils are phagocytes. They are the most ubiquitous or most common of these white blood cells. And their primary purpose is really to gobble up bacteria. So they are on defense, usually circulating, surveilling blood, but they also can escape blood and get into the tissue layers for the purpose of looking for things to gobble up, to phagocytize, and especially bacteria. Basophils are major components of the inflammatory response. They produce histamine, they produce heparin, so they are primarily going to be involved with that. Lymphocytes, I'll do a whole thing on lymphocytes because obviously lymphocytes differentiate into T cells and B cells, and then that even gets even more complicated, especially with T cells. But in general, B cells and T cells are our lymphocytes, okay? And there's also natural killer cells, another one to talk about as well at some point. Monocytes are these very large cells that basically mature into macrophages whenever they get the signal, the cytokine signal to do so. And their purpose, obviously, as a macrophage is to phagocytize cells, cellular components, cellular fragments, or other things like bacteria, like you would see with neutrophils. Thrombocytes, these cell fragments, particularly when we talk about them, they're involved with hemostasis, but again, they also produce some growth factors that are very important as well. All right. Now, the other thing I wanted to point out is obviously cell morphology. If you notice, obviously erythrocytes, they don't have a nucleus. They lack a nucleus, so they don't stain with the hematoxylin. They have that wonderful little kind of almost like a donut shape. They do have a bicave indentation, kind of in that central component. Eosinophils have this bilobed nucleus. As you can see, it kind of like taffy. If you pull taffy in the middle out to the side, you kind of have these two blobby ends and this stretched end in the middle. That's my analogy that I'm going to use here. So eosinophils kind of look like if you're pulling taffy apart. So they have this bilobed nucleus typically. Neutrophils have multi-lobe nuclei. In some cases, it almost looks like you have separated pieces of the nucleus. Just recall that you're looking at a two-dimensional shape when these are in reality three-dimensional shape. But neutrophils do have multi-lobed nuclei, as you can see here. So lots of little kind of bubbling components of the nucleus. It's not asymmetrical at all. Basophils do have a nucleus. I purposely drew this way because very often in my experience when I've looked at different types of slides, basophils, most of what you're seeing are going to be these very dark basophilic vesicles, these granules within the basophil. But I do have an image where we actually can see the nucleus as well. So it's not to say that this thing is devoid of a nucleus like a red blood cell. It does indeed have a nucleus. But very often the nucleus is being kind of masked from all the dark basophilic granules or vesicles that are being stained with the hematoxylin. Lymphocytes have very large nuclei. Often lymphocytes can be mistaken for basophils because in some cases the nucleus looks like it's taking up the entire mass of the cell. And indeed the nucleus in terms of ratio to the plasma membrane and the cytoplasm, the nucleus is very large, or I should say in comparison to the cytoplasm, not the plasma membrane. So it is pretty large. Monocytes. Monocytes have a little bit of an indentation. In some cases, you might mistake them for something like an eosinophil. You would think, okay, it looks like it's very bilobed. So it has a little bit of an invagination or an indentation in the nucleus. It also has a very large nucleus too, compared to the cytoplasm. And they're going to be the biggest cells that you're going to be looking at. So monocytes are the largest of these white blood cells. They're very large cells. Lymphocytes and monocytes, again, are agranule, so it's not to say that their cytoplasm doesn't have anything in it, but it's not big things like these big vesicles that are going to be carrying certain important parts for the function of that cell. So most of what you're seeing, obviously, is something that doesn't look really granule at all, or it looks very agranule. Okay? And then thrombocytes, as we'll see, they're often really dark purple, little small, kind of either symmetrical or asymmetrical looking dots in the blood smear. All right, so let's look at a general one here. And I'm borrowing from Duke University School of Medicine. Duke University School of Medicine and University of Michigan, as I've used in the past. These are wonderful virtual histology slides that I recommend using. There's other ones out there as well, but they're very fantastic. I've become a little bit more partial to Duke, and Duke indeed uses some of University of Michigan's slides, their web slides. But I've been having experience with University of Michigan where it takes a while to focus when you zoom in. And that can be a little bit frustrating, especially when I'm teaching my virtual anatomy class and I'm trying to show these different things to my students, and we're having to wait, in some cases, maybe 2 minutes for it to focus. So not sure what's going on with the University of Michigan. I used to praise it like crazy, but unfortunately it's lost some important functions like the drawing tool, and it also, for some reason, has issues when it comes to focusing. So I'm using Duke University School of Medicine's slides from their website. I recommend, especially as you start to practice, as you should, to help you prepare for your histology exam, histology lab exam, or maybe the histology component of your lab practical for A&P. These are great websites to go look at these different things and really kind of practice and get a good feel of what you're looking for. Okay? All right, so that's my spiel for Duke University. Okay, so we have a couple things here. And as always, I always recommend to my students that you start with the lowest magnification and then zoom in. This is especially true when you're looking at tissues and you need to differentiate between tissue layers, and even within those tissue layers, maybe specific components of those tissue layers. Now, it is worth noting that when you're doing a blood smear, everything is going to look mostly pinkish, a bunch of little pink dots. So not at the highest magnification at 40 times, not exactly something you're going to be able to differentiate, but like, oh yeah, I can really distinguish these different types of cells. But as you start to zoom into, say, maybe 20 times magnification, you'll start to see dark purple looking things among this sea of these pink roundish cells. And again, recall that most of blood is erythrocytes. Most of the formed elements, I should say, of blood are erythrocytes, these red blood cells. So they are 99.9% of the cells you're seeing. But as you start to zoom in, you're going to start to see some nuclei that are being stained by the hematoxylin. So you can actually start to see some purple things. And if your lab obviously is going to be telling you to look for white blood cells and then distinguish what white blood cells you're looking at, this is a good way to be able to pick where the white blood cells are in that field of view, and then further zoom in to really look at that white blood cell and see what type of white blood cell you're looking at. Now, I chose this view here about 20 times so that we can see that there are different types of cells here, these different types of white blood cells. All right, there are 3 different types of white blood cells here. I'm actually going to kind of pause a moment and see if you can think what 3 general white blood cells we're seeing in this image. All right, so let's talk about them. What are the things we're seeing here? So clearly this is a white blood cell. It has a nucleus, unlike the red blood cells. all of these being red blood cells. This has a nucleus because it is being stained by the hematoxylin. And again, nuclei stain dark purple, blue because of the hematoxylin of the H&E stain. This one also clearly has a nucleus, as does this one, this one, this one, and this one. Sorry, let me increase the size of my pen here. Might be a little bit better. Okay. So we can see, obviously, there's some differences in morphology. All right. And I'll go ahead and point out, before I start to actually point them out, that the three different types of white blood cells that we can see in this image are going to be monocytes, neutrophils, and lymphocytes. So those are the three types of white blood cells I see in this image. Neutrophils are, again, the most common. Roughly about 70% of the white blood cells you're looking at are neutrophils. Neutrophils have a granule texture to it because they are granulocytes. But it is worth noting, unlike eosinophils and basophils, those granules don't stain as dark or as thick as eosinophils and basophils. But more importantly, if you're trying to distinguish a neutrophil from, example, an eosinophil, which would be the thing that looks closest to a neutrophil, as I just mentioned, the granules of neutrophils are smaller and tend to not stain as thick and as dark as an eosinophil. But the other important thing is the multinuclei. As you can see, this clearly has multinuclei here, as does this one and that one. So all of these, I'm going to put N's, are neutrophils. All right. Now, the other two that I want to highlight here are going to be this one and this one. All right, these cells. These cells could be mistaken for basophils, especially if you're looking at it from this magnification. You would really want to zoom in to be able to confirm, know for sure what you're looking at. This one especially could be confused for a basophil at this magnification. But this one, I can see a little bit of a plasma membrane on the right-hand side of the cell. And therefore, if I can see a little bit of a plasma membrane that's just a kind of a lightish blue color, I would say, okay, that most likely is going to be a lymphocyte. And indeed it is. Lymphocytes have very large nuclei. So that is a lymphocyte. And this one, this mystery one here, is also a lymphocyte. So I'll put a little L for lymphocyte there. Again, if you're not sure if that's a lymphocyte or a basophil, my opinion, the two that are also easily confused, just like eosinophils and neutrophils can be easily confused with one another, you'll want to zoom in to a higher magnification. And you may actually want to use oil immersion and go all the way up. Okay, so it's good to make sure you're looking at exactly what you're looking at. The last one I want to point out here, obviously, is this one in the upper kind of left-hand corner, close to the left-hand corner. This one has that distinguished, first, it's a very large cell. If you compare it, especially to something like a lymphocyte, this thing is mega. The other thing I want to point out here is the very large nucleus that has a little bit of an indentation, as you can see. Actually, it almost looks like it has two. So as I mentioned before, if you have something that looks very agranule, it has a indentation in the nucleus, a very large nucleus, and is very big. Those three kind of clues, those three giveaways tell you that what you're looking at is a monocyte. All right, so that is a great example of a monocyte there. All right, so let's look at some other examples because we still have two other cells, actually three, that we haven't looked at yet. So this one is an interesting one. All right, this one can be a little bit confusing. Might be a neutrophil, might be an eosinophil, might be a basophil. So how do I tell the difference? What should I look for? Recall that I mentioned that the neutrophils are granulocytes, but the granules tend to stain first. They tend to stain, I shouldn't say they tend, they do stain with the eosin primarily. So most of what you're seeing are going to be eosinophilic vesicles. But it's also very light, such that you can see a little bit of some granule texture to it, but it's not very thick. This one is very thick. This one has lots of very thick granules, as I can just see here. So what about an eosinophil? Eosinophil, remember it's in the name. Eosinophils are eosinophilic. They are eosinophilic. They get that term because they love the eosin stain component of the H&E stain. And eosin stains components of cells a reddish color. All right, reddish, pinkish, maybe sometimes orange. And indeed, I do see, obviously, some pink, reddish hue within the cytoplasm of this cell. All right, so that could be an eosinophil. Eosinophils, aside from the nucleus, really do not stain with the hematoxylin. So only the nucleus of the eosinophil is really going to be staining kind of in a very distinguishable way, is the only thing that's really going to be staining with the hematoxylin. Okay, but this clearly has some granules that are staining really dark, as we can see, especially on the periphery here, close to the plasma membrane, and even throughout, you can see lots of these dark stains. This one really does stain, has these basophilic vesicles that stain with the hematoxylin. So this one, therefore, is definitely a basophil. All right. And you're probably thinking, well, you said basophils usually have this, mostly all you see. You don't see the nucleus, and mostly all you see are these dark basophilic vesicles that are staining. Very true. But as I mentioned before, it does have a nucleus, and sometimes you can see the nucleus. And you can even see some of the other components of the cytoplasm being stained with the eosin of the H&E stain. So I chose this one. Actually, Duke University specifically has this as an option on their website, which I recommend looking at. That can be a challenging one because it could look like an eosinophil. You might confuse it for an eosinophil. So this is a good one to definitely look at. But obviously, when you look at your own slides or if you practice using some of these websites I recommend, you definitely should kind of really look at the cell and see if that is indeed what you're looking at. So again, it kind of looks like an eosinophil, but we have these really dark vesicles that are basophilic. So therefore, this is a basophil. The other things I want to point out that I mentioned before are these dark purple little small things, the smallest cellular things that you're going to be seeing in here. These are going to be the platelets or the thrombocytes. All right, so the only thing you really see stained is the components of the cell fragment that are basophilic. So often the smallest things that are usually like a dark purple color are going to be your platelets. All right, so last thing we haven't seen obviously is going to be our eosinophil. We have two white blood cells in this image. Both of them are granule cells. As you can see, they have a granule look. This one definitely has some granules, albeit not very many or kind of faint looking, but distinguishable. This one definitely has more of a granule texture, or more of a granule look. All right, so how do we distinguish these two? Because these are clearly two different types of white blood cells. So recall I mentioned that neutrophils are granulocytes. They're multi-lobed, and the granules tend to not show up as distinctly as in an eosinophil or a basophil. And these do mostly stain with the eosin stain. So most of these granules within this neutrophil are going to be eosinophilic. So the eosin stains them. So definitely, as you can see here. So this other one, as we can see, does it have vesicles that are basophilic, like we saw with our basophil? So lots of really dark, round, even thick, round circles, vesicles, granules in this cell? Or does it look more pink, more reddish, more orange color? So hopefully everybody was saying, it looks more pinkish, reddish, orange color. Well, it looks more like a weird dark pinkish color. So this is definitely an eosinophil. All right. As we can see, it has that bilobed nucleus, which is staining with the hematoxylin, but it also has this dark pinkish cytoplasm that looks very granule-y. I keep wanting to make granule-y a word, but if it isn't a word, it is now. So it looks very granule-y, but it's not dark basophilic granules that we're seeing. These are eosinophilic granules that we're seeing. So therefore, this tells us that we're looking at an eosinophil. Great. All right. Last thing I want to bring up here is this cool image. This is actually the serosa of the colon of a monkey. And I brought this up because we have some really cool things in here. The first thing I want to highlight is we have some blood vessels. Here's a blood vessel. Here's a blood vessel. Okay. You can see obviously the smooth muscle within these blood vessels. And you can also see something else that I think is important that I will definitely talk about at some point. But these really kind of flattish looking cells close to the lumen or the inside of this blood vessel. All right, I'll get to those here in a second. But how do I know this is a blood vessel? Well, as you know, red blood cells have this biconcave shape. They have no nucleus, so therefore they only stain with the eosin. So as you can see, there's lots of cells packed in this blood vessel, these red blood cells that lack a nucleus. They don't stain with the hematoxylin. They do stain with the eosin, and you can even see some of that concave shape or biconcave shape of the cell. So that automatically should tell me that I'm looking at a blood vessel. Also, these flat irregular shaped cells close to the inside of the lumen are in contact with the lumen. So if we think this is the lumen or the opening of the blood vessel, we have these on the kind of margin of that lumen, we have these flat nuclei. These are endothelial cells. All right, endothelial cells are a special type of epithelial cell, and these are squamous cells. Endothelial cells are squamous cells, and I will do a video when I talk about epithelial cells and how to distinguish between a cuboidal versus a columnar versus a squamous, and even the different arrangements of those things. But endothelial cells, squamous cells tend to have flat, irregular shaped nuclei, because it is a flat, irregular shaped cell, right? It's a squamous cell, which is a flat, irregular shaped cell. So the cells, endothelial cells that line the blood vessel are squamous cells, and you can see they have these flat, irregular shape kind of nuclei. Some things that are inside these blood vessels, aside from the red blood vessel, sorry, let me erase some of this stuff here, that I want to point out are this cell. Get rid of this one here too. And these cells. These are eosinophils. Now, I don't know what was going on in this poor monkey before, unfortunately, it was used for the purpose of making these slides. But the fact that we're seeing a few eosinophils in here is kind of interesting because eosinophils are essentially the second least common of these white blood cells. And here we have two nice images of eosinophils. You can see it has these really nice granule texture that is being stained with the eosin. Okay. Now, the other really interesting that's going on in this poor monkey specimen here is within the tissue itself. So these are our blood vessels that are essentially embedded within the tissue layer. And if you're not familiar with this term yet, we tend to talk about the tissue layer, the space and things within the tissue layer as the interstitium. So I'm just going to put an I here. So all this area here is the interstitium of the serosa, the outer layer of the colon in this case, but also other components of the GI tract. And we have lots of cells in this area that look very similar to each other. And hopefully you're comfortable enough already to know what these cells are. But I will do a dramatic pause while you think about what these cells are. What are these mystery cells? So, every one of you is saying neutrophil, and bravo to you because you are correct. These are all neutrophils, all right? Neutrophils have the ability to escape blood and go into the interstitium, and indeed that is an important feature of a neutrophil, particularly under the influence of this cell right here. This cell is a mast cell. Mast cell. Mast cells tend to exist in the interstitium. It often is described as looking like a fried egg or like a very granule looking fried egg. Kind of looks like that, definitely. The nucleus looks like a nice yolk. You can see it has all these wonderful little eosin stained vesicles. Mast cells release histamine and cytokines. So the histamine basically causes vasodilation. The blood vessels in the nearby area will increase the size of their lumen, the diameter of their lumen. And also the space between these endothelial cells gets wider. So normally, small things are able to escape blood and go into the interstitium because this is what we want. We want gases to be able to cross these barriers between the interstitium and blood. And even some small solutes like glucose and other things, also amino acids, these things can escape blood and go into the interstitium and vice versa. But large things, particularly cells, don't necessarily get out of blood and go into the interstitium because, at least in these continuous capillaries, the space between the endothelial cells is very small. It's not big enough for them to squeeze through. Under the influence of histamine, the spaces between the endothelial cells widens. And this allows neutrophils and monocytes, which become macrophages, to escape blood, go into the interstitium, especially with the express purpose of looking for particularly bacteria or other things in the interstitium that's causing infection so that they can phagocytize, so that they can destroy it. So here we have lots of neutrophils in the serosa of the colon of this monkey. Not exactly sure why, but it is worth pointing out, obviously, that we do see lots of neutrophils here. So neutrophils can escape blood, especially because mast cells produce histamine and cytokines. The cytokines attract the monocytes and also attract the neutrophils via chemotaxis. And also the histamine that's being produced increases the space between the endothelial cells of the blood vessels so that neutrophils and monocytes can squeeze through and get into the interstitium. All right, that's probably good enough. So hopefully you found this useful. Definitely recommend to go to these websites to help practice, obviously, for your upcoming lab practical or your lab handout, whatever it is you're working on at the moment. And I'm going to continue doing lots of histology because right now it's something that I'm very keen on and very interested in. But obviously I'm going to continue. Primarily I focus on anatomy and physiology because that is mostly what I teach as an instructor. But at some point I will also start to include some biology material as well because I also do teach biology. But all of it is fantastic. That's all I got to say. So good luck, happy studying, and thank you very much.